Hi, everybody. Welcome in. Welcome to another OpenTrons webinar. We're going to take 5 minutes to let the participants trickle in. And after about 5 minutes, we're going to go ahead and get started. Just a reminder that this webinar is going to be recorded and the slide decks will be sent to the participants at the end of the webinar. But for now, we're going to let some participants settle in and we'll get started in a couple minutes. So just settle down for a little bit. Thanks for joining. Again, for those who are just joining, I just wanted to repeat myself. We're going to take a couple of minutes to let the participants settle in, and we'll get started in a couple of minutes here. Thank you for joining, Joseph. All right, it looks like the participants have settled in, so we're going to go ahead and get started. Again, welcome to another OpenTrons webinar, and today's topic is how to choose the right automation for your lab. So this is primarily focused towards those who are new to automation, scientists and lab managers who are considering using liquid handlers in their laboratory and what you should consider before moving on to the purchasing process. Just to give you guys a little bit of background about myself, I've been in the biotech and automation industry for about 7 years, going on 8 years. Before working at OpenTrons as a product manager and dealing with commercialization, I was also a product manager at Zymo Research Corporation in Irvine, California. And I also worked at Eppendorf before this as an application scientist. And I primarily deal with commercialization of our new product, the OpenTrons Flex. And I've also commercialized several liquid handling kits in the past. So the objective of this webinar is to familiarize yourself with different types of levels of lab automation and also understand considerations of choosing the right liquid handling equipment before moving on to the purchasing process. So here's a quick agenda. We'll do a quick icebreaker / intro to liquid handling automation for those who are not familiar. We're going to move on to the benefits of automation, and then we're going to dive deep into the considerations for automation in your laboratory. And then I'll go over a couple of liquid handling case studies and how we come up with an automated solution based on those case studies. After that, we'll do a quick 10 to 15-minute Q&A after the webinar for any questions that you may have. Cool, let's get started. And again, this webinar is being recorded and it'll be sent to you after this webinar ends. Cool, so let's break the ice here and talk about liquid handling and the evolution of liquid handling, right? So microliter level dispensing was really started to be needed in gas chromatography applications. And because of this, the first ever microliter syringe was developed in the 1950s. 10 years later, the first ever spring-loaded pipette was commercialized by Eppendorf. And in the 1970s, when microprocessor technology started to advance, we started seeing electronic pipettes. So these manual pipettes that you see in your standard everyday lab, and Hamilton launching the Microlab M, which is one of the first electronic pipettes. In the 1980s, Hamilton and Tecan actually collaborated to release something called the Amica system. And I would say this is the first ever benchtop automated liquid handler. And it was really a starting point to really to evolve the liquid handling space. And then moving on to today, and I'll just move to the next slide here. But today we have a plethora of providers of liquid handlers. We have a ton of pipette options, single channel, 8 channel, 12 channel, 96, 384. Not just talking about Eppendorf, but just the industry in general. We have robotic grippers that can move plates from position to position. And we also have added functionality to your liquid handlers now in the form of modules like a heating and cooling and temperature control, shaking, as well as on-deck thermocycling for your PCR reactions, and much, much more. So this is kind of a bit an overwhelming slide, but the point of this slide is really to show there are a ton of different liquid handlers out there. There are entry-level liquid handlers and aliquotters. There are medium benchtop liquid handlers, high throughput large liquid handlers, but we also have those niche liquid handlers, like ones that are focused on regulatory compliance, one that are application focused for one certain application, ones that's focused on the software, OEM. So how do we choose between all of these different liquid handlers? So hopefully I can help you out today. Great. So getting into the benefits of automating your pipetting workflow, right? There are a couple of obvious benefits like saving time. And when we talk about time, I really am talking about walkaway time. Sometimes a liquid handler may not perform as fast as you may do it manually, but this just means that while this is performing this function for you or your experiment for you, you can get up and do your other type of scientific work at your workbench or at your office or anything like that. Also, high throughput processing, you tend to save time once your sample size grows to, more than 96 samples, or you're processing a whole plate at a time. Another benefit is really reducing cost. Although there's a high capital investment in investing in liquid handlers, we do have long-term savings, and we'll be talking about that more. Increase accuracy and reproducibility, right? If a robot is pipetting for you, it will be extremely accurate and reproducible, preventing human error. And this leads to having to redo your experiment if you made a mistake in the middle of this long, complicated protocol, right? Increased throughput, right? Doing an experiment manually really inhibits your throughput. People can't really do more than, 8, 16, 24 samples without it taking an extremely long time. So liquid handlers shine in this type of space. Increasing data integrity. So a lot of liquid handlers on the market can be integrated into electronic lab notebooks and LIMS systems. And you can, and audit trails, you can track who signed in, who changed the protocol, what type of data that liquid handler produced, and store that data for the long term. This is all completely automated. And then finally, reduce physical strain. Sitting on the lab bench every day for 8 hours a day will definitely strain your hands, cause carpal tunnel, and this reduces that. Now getting into the considerations for automation, and this is what we're primarily going to be talking about for this webinar. So we're going to talk about costs, cost considerations. We're going to dive deep into data integrity, data quality, modularity and flexibility, sustainability and consumables, throughput and scale, and then we're going to go into safety as well. So these are all the considerations we'll be going through. Just a quick call out. A lot of this content is really based off of a really great SLAS review article that was released last year. If you have time after you receive this slide deck, I recommend you go through this article because it really talks about a ton of these considerations that I'll be talking about today. Awesome. So let's dive into it and first talk about costs. When considering costs between manual and automated processing, obviously we have the cost of labor if you're doing it manually, the cost of PPE, and then especially human error. If you make a mistake, you'll lose money on reagents. You'll have to redo the experiment over again. However, you do save on consumables. Consumables are generally higher when in an automated system, and also the hardware, right? Manual pipettes are generally lower cost than a full liquid handling machine. When going into the automated system, in terms of costs, yes, the hardware can have a high capital investment, but you also have to invest in service, things like preventative maintenance, installation, and again, that high consumable cost. But you do save money on labor. You save money on tip consumption, going back to human error. If they make a mistake, you might have to, use double the amount of tips when doing one experiment. And that can be prevented with the liquid handler. Diving deep into costs, I would say there are three different levels of costs with liquid handlers. You have your entry-level liquid handlers, which are more on the inexpensive side, I would say, and it's really dependent on the size and available features of the robot. The ones that you see on the left here are more for simple liquid handling workflows. They have minimal deck positions. However, if your experiment is really simple, like a simple PCR, this might be a liquid handler for you and won't really cost you that much. For more complicated workflows, if you want to, automate something that needs heat control or a thermocycler, consider a medium benchtop automated liquid handler, especially if your throughput is not that high. You can still do those complicated workflows at a pretty affordable price. And then finally, you have your big high throughput automated liquid handlers that are meant for doing complicated protocols in high throughput, and it has a ton of advanced features like barcode scanning and plate shuttles for increased throughput, as well as like specific incubators for specific applications. And these are primarily in the high range in terms of costs. So consider these three different brackets of costs. Excuse me. Now, the next consideration we're going to talk about is throughput, which kind of goes hand in hand with cost. So when considering throughput in a manual process, if you have a very low throughput, which means you're processing a small number of samples every day, then I would say that the manual process excels compared to the automated process because, let's say you're processing 8 samples a day with your hand, the cost savings and the throughput may not be worth it to invest in a liquid handler. But this may be limited by work hours and human fatigue. Yeah. And then moving on to automated liquid handlers, they're accurate and repeatable. You have fewer errors, which means you can do more runs. And the primary reason is you can increase efficiency tenfold more. So what I mean by this is, if you process 8 samples of PCR a day or a full plate of PCR a day, a liquid handler could probably do 10 plates a day. So consider that if you want to scale up your throughput in the future and you don't know how to accommodate this solution, consider a liquid handler if your lab plans to grow. So here's also a breakdown of entry-level liquid handlers, medium benchtop liquid handlers, and high throughput liquid handlers, and the type of throughput they may be able to accommodate, right? If you're working with less than 96 samples and you don't plan on scaling up, I would say consider using an entry-level liquid handlers, those smaller liquid handlers with minimum deck positions and that are affordable. And this is if you're running your protocol maybe once or twice a day. If you want to process up to 96 samples and more complicated workflows, I would get something like a medium benchtop automated liquid handler. It still fits on a benchtop, and you can do all your complicated workflows up to 96 samples most of the time. And then finally, you have your high throughput liquid handlers. This is if your laboratory wants to really scale, if you want to run end-to-end processes every day, all day, at max capacity, consider this type of market. And throughput, again, is primarily dependent on deck space for labware. I would say specifically tip space, right? You only have a certain amount of deck positions to store tips, and usually you want to change tips for complicated workflows to prevent cross-contamination. Cool, and this is a really great breakdown of the capabilities of entry-level, medium benchtop, and high throughput liquid handlers that are application specific, right? So breaking this down, if you're doing something like sample prep, filling tubes up, going from plate to plate or tube to plate, a entry-level liquid handler can definitely process up to 96 samples, while medium and high throughput liquid handlers can process multiple plates at a time because the application is so simple. Moving on to PCR, again, PCR is quite a simple process. If you want to process up to 96 samples, those entry-level liquid handlers can definitely do that. While the medium and high throughput can do multiple plates at a time. Getting more complicated, talking about something like nucleic acid purification and bead cleanups, right? I would say an entry-level liquid handler is limited to between 8 and 16 samples, maybe 24. But most cases, it's between 8 and 16 samples. But if you have a medium benchtop liquid handler, you can process a whole plate at a time. And if you have a high throughput liquid handler, you can definitely do more than one plate in one run. Getting even more complicated, talking about next generation sequencing library prep, something that has multiple PCR steps, multiple cleanup steps. I would say your entry-level liquid handler can process only parts of the step at the lowest throughput possible. So let's say, for example, only doing the tagmentation and first PCR amplification, right? That's something a small liquid handler can handle in 8 samples. If you want to scale up or if you want to do an end-to-end process, you definitely need to invest in something like a medium or high throughput liquid handler. And then finally, ELISA. ELISA is actually not as complicated as NGS in my opinion. It does require a lot of reagents. It does require a lot of incubations. So in terms of plate filling and washing plates and stuff like that, your entry-level liquid handler can probably process one plate at a time, while your medium and high throughput liquid handlers can exceed that one plate threshold. Just a little guide for you guys when you guys receive the slide. Great. So let's move on to the next consideration, which is modularity and flexibility. So obviously, in the future, your experiment may change. The number of samples you process may change, right? And in a manual process, it's very easy to change your experiment because you're controlling your own hands. But this does require expertise, right? It does require a scientist's expertise to change the protocol. However, it is quite flexible. In an automated perspective, you can easily scale up your application if you anticipate growth, right? If you set up your application to process 8 samples at a time, it's quite easy to say, now I want to process 48 samples because my laboratory grew. And that's when liquid handlers shine, really. Programming and error handling may require technical support, which is definitely a drawback, right? If something goes wrong, you may need to contact customer support, which can hold you up. And it could take months to change your automated process if you are using an application scientist or somebody that's writing the protocol for you, compared to a manual process where you just need to change, the protocol manually with your hands, right? So I would say if that's important to you, I would consider a system that you can program yourself. So a lot of liquid handlers out there have easy to use graphic user interfaces where you can program your own experiments. This is quite a common theme in the liquid handling space. And if you pair this with very strong applications and technical support, you shouldn't have that many issues. Next, I would say consider being an expert at the manual process, right? If you're going into liquid handling automation, lay out everything you need on the table for your manual experiment and match that with the type of hardware that you would need for your automated liquid handler. This is a great example, right? You have a manual PCR setup on 24 reactions. You do cycling on an external thermocycler, and it's something that you want to scale up in the future. Something that you want to, level up to 96 samples or something like that. So, obviously you have the base unit liquid handler. If you're doing a PCR reaction, consider using a single channel or an 8-channel pipette based on your preferences. You have a robotic gripper that can move the plate to your thermocycler, and you need the thermocycler itself. If you want to continue to use your own thermocycler, that's totally fine. It's quite common. You can just do the liquid handling portion on the liquid handler, and then once the protocol is done, you can just take that plate and manually move it to your preferred thermocycler. Great, next slide. Next consideration, we're going to talk about data integrity. So obviously, in manual processes, it's extremely variable, right? And not just from run to run on the same person, but from person to person. They may use their pipetting techniques may differ depending on that person, and that yields different results, and it may skew your data. So random manual errors, and then fatigue can also lead to drift, right? If all of you remember sitting in the clean room doing a PCR and doing 96 samples one at a time and doing your serial dilutions, you can lose track very easily. Trust me, I've done it tons of times. With an automated liquid handling, that is all avoided, right? It's accurate, repeatable. It actually detects errors, right? If it doesn't detect a certain liquid, well, most liquid handlers do detect errors. If it doesn't detect a certain liquid, it'll throw an error. If it crashes with another piece of labware, it'll throw an error. So this is extremely manageable if you have something called error recovery or error processing. And also, some liquid handlers are a closed system with better environmental conditions. And we'll get into that a little bit later when we talk about HEPA UV and sterilization and stuff like that. Okay, so getting into data integrity, I want to take the time to talk about liquid classes. So what liquid classes are is all these different small settings that you program in the liquid handler that will optimize your pipetting. And different liquids with different viscosities need their own liquid class or set of parameters to optimize the aspiration and dispensing. So some things that you need to consider, the quality of the tip. I usually recommend using the vendor approved tip. A great example is at Opentrons. We highly recommend you use our pipette tips with our liquid handler because our scientists did all the liquid class testing on those tips. Excuse me. Pre-wetting. Pre-wetting is something that you can do to increase your data integrity in your liquid class. Usually for stuff like that causes evaporation, like ethanol, very volatile, and liquids that cause vapor pressure. And if you aspirate the liquid and dispense the liquid before performing your liquid handler, it'll reduce this. So consider that as another setting. Your pipetting mode is something you should also consider. You have forward pipetting, which basically pushes the piston forward and forces the liquid out with air. You have reverse pipetting, which pre-aspirates and then has no blowout. So it'll pre-aspirate and it'll have an additional piston movement to accurately pipette your liquid and then blow out. If you have, a little bit of liquid left in your tips, you can program a blowout, which will blow more air to get all that liquid out. And then aspiration and dispensing settings, you have, tip depth, how low you want to go into your liquid. You have the angle of where you want to submerge your pipette. The speed, right? Aspiration and dispense speed matters a lot. You probably want to aspirate very slowly in a viscous solution. Dwell time and setting time, that is how long the tip stays in the liquid after an aspiration or dispense step. That can optimize your settings. Withdrawal speed, how fast you withdraw from the liquid, and then stop back volume. And that's basically the volume of air dispensed when you're doing repeater pipetting. These are all things that you must consider when optimizing your liquid class. Cool. So talking about data integrity and going into like certain parameters, there are also miscellaneous parameters that can affect data quality. So temperature is a big one, right? Warmer air decreases your estimated volume, and we usually recommend working at ambient temperatures or between 15 and 30 degrees C. Humidity is a huge one. We have this issue all the time, especially with pipetting very small amounts of liquid, something like 1 microliter. If the humidity is different, the liquid class will definitely change, and that's something you need to consider. Recommendation is between 45 and 75% humidity. If you're working in something with higher or lower humidity, consider adjusting your liquid class. And then altitude. Altitude is also something that can the pressure differential will definitely affect your liquid class. So you should always calibrate at altitude. Great. Moving on. So more liquid properties. So volatile liquids. I mentioned this before. If something creates a lot of vapor pressure, it can cause dripping. It's very common in ethanol aspirations with liquid handlers. So definitely consider this. We recommend pre-wetting the tips. We recommend air gaps to prevent dripping. When you're dealing with viscous liquids can cause air bubbles, and they can also stick to the tip very easily. So we recommend slow aspiration and withdrawal. We recommend using low retention tips, maybe even wide bore tips if your liquid is that viscous. Trust me, I've worked with E. coli before after we've lysed it, and it's basically a very viscous, snotty type of liquid. And then finally, surface tension, right? Surface tension is pretty important. It can cause foaming and droplet formation. So again, we recommend using low retention tips, decrease your pipetting speeds, and minimize the setting time. Great. Okay, next consideration is safety or occupational safety, right? The first thing we really call down occupational safety is repetitive stress injury. For those of you who sit at a lab bench every day for 8 hours a day, you can prevent carpal tunnel or any type of injuries and any type of fatigue by investing in a liquid handler. Also, it leads to poor posture, right? I have pretty poor posture because I used to be a scientist. And then exposure to harmful chemicals. If you're working with like phenylchloroform or something like that and a spill happens, that's quite dangerous. And a liquid handler, you would rather have it spill in a liquid handler than your hands, if that makes sense. And in an automated perspective for occupational safety, we have built-in safety features like brakes, emergency stops. It definitely decreases foot traffic for emergencies because it'll be on a bench. And as well, you have hands-free operation in a closed, and if you want, a vented system, something like a HEPA UV module. So let's talk about that, right? We have different things that you can add, or the general liquid handling market generally has things to minimize or maximize safety, right? So the first thing I'll call out is HEPA filters. So I just want to say that HEPA filters really protects your samples and does not protect the user. Because the way it works is it filters contaminants through the filter and the air actually gets dispelled out of the system. So if you're working with something like like a COVID virus or something, don't expect an HEPA UV filter to protect you in that way. So I would say it protects your samples and not the user. UV sterilization. So if you're working with bacteria, some HEPA UV modules or HEPA modules have UV add-ons that you can turn on either after your run, before your run, or in between your run. And this sterilizes the work table between runs. And it really just breaks down the DNA and RNA of microorganisms. This is very common in microbiomics experimentation and microbiomics automation. And then finally, if you want to protect yourself, biological safety cabinets, a lot of small liquid handlers can fit in biological safety cabinets. So if you're working with harmful chemicals, consider something small or consider investing in a custom biological safety cabinet that can surround your liquid handler. Great. So let's get into documentation, and this is really calling out, electronic lab notebooks, LIMS systems, and those type of electronic documentations that you may need for your laboratory, because some laboratories do require this, right? So in a manual process, data documentation is difficult, right? It's a manual process regardless. Even if you have, a manual or a standalone PCR machine, you need to take that data, and it can be automated into a LIMS system. However, this is all a manual process going from machine to machine, right? And it's difficult to detect pipetting errors in the workflow, right? If you have something like a LIMS system or an electronic lab notebook, it can document it can document any changes that were made in the workflow. It can document any errors that were brought up with error recovery and whatnot. And then exposure to harmful chemicals. Automated on an automated system, some automated systems have automated barcode scanning, so you can track your samples from beginning to end, and see exactly what happened to those samples through your experimentation. Again, automated error handling and liquid detection, right? If you don't detect a liquid in the middle of your protocol, it'll throw an error, and that error will be logged in your electronic lab notebook. It's a much more streamlined process. And again, audit trails, I called this out before. And the log files are freely available with most liquid handling systems, and that could be incorporated into your LIMS system or electronic lab notebook. Great, moving on. So considering documentation, if this is important to you, I would consider software that supports liquid level detection, absolutely. So it can throw errors. So that's the other point, error detection. Another call out is CFR Part 11 compliant. So this is a term that you probably heard before, and it does require tracking samples, tracking user logins, tracking audit trails, electronic signatures, and whatnot. This is very common in the pharmaceutical and diagnostic space. So if you're a lab manager in a pharma lab or a diagnostic lab, consider having this either built in or having your tech team strategize to be compliant with CFR Part 11. And then again, is compliant with particular regulations, usually for in vitro diagnostics, right? Offers GMP and GXP compliant software. So this is a little different from CFR Part 11, but it's basically good manufacturing practices and good laboratory practices that have quality control procedures, documentation on those quality controls, like IQ, OQ is very common, right? And then can interface with LIMS and ELN systems. So some are already built in and can easily interact with these LIMS or ELN systems. And if not, consider a automated device that is open source, so you can funnel that data into that system. Great. Okay, moving on to one of the last considerations, sustainability and consumables. So in a manual perspective, more mistakes obviously mean you're using up more consumables. However, you have more flexibility to choose the type of consumable, right? A lot of liquid handlers are tied or optimized for one type of consumable. So you can be very consumable conscious when you're working manually. And it is cheaper on average. Manual consumables compared to specific automated consumables are cheaper. In an automated perspective, um, usually consumable packaging promotes more waste. So if you're very green or very environmental friendly, this is definitely a drawback. You have higher dependency on a single supplier, right? A great example is pipette tips. We recommend using our pipette tips. If you want to use a different pipette tip, you may not have the same level of data that we promise. And then you usually save tips in an automated perspective because if you make a lot of mistakes or if you need to redo your experiment manually, this doesn't usually happen in an automated system. They also have automated tip washing and metal tips in some systems. So if that's something you want to get into, I would consider looking into that. Okay, I think that we went through all the considerations. So the next part of this is I'm going to talk about two case studies, very common scenarios that happen at Opentrons all the time. And I'll tell you guys how we deal with it in an automated perspective, right? So the first example, obviously all of you know about COVID-19 and the pandemic outbreak. So there's a laboratory that is doing COVID monitoring and sequencing, right? They want to sequence for specific mutations over time, and they usually process 8 samples a day manually. And they're using Illumina COVID-seq, which is a generally simple seq library prep protocol. And their challenge is they want to scale. They want to increase their capacity to 16, eventually 24 samples daily. They're growing. They need to do more tests. They need to do more monitoring. And they currently use a PCR-free room. So the way their workflow works is they do the first half of the library prep outside and the second half in a PCR-free room to prevent cross-contamination, right? Usually post-library cleanups are done in a PCR-free room or a clean room. They have no prior experience to automated liquid handling system, and they had very limited space. So especially in their sterile areas. Yeah, and this is kind of the scenario, and how would you solve this type of scenario? So I'm going to go to the next slide, and this kind of shows the solution here. So this is a very common solution we recommend to our customers, right? We recommend using a small or medium benchtop liquid handler. And we recommend purchasing more than one for your non-clean room and one for your PCR-free room. Because this is a medium or small benchtop liquid handler, space is less of a concern. You don't have to spend, hundreds and thousands of dollars on these huge devices and place them in 2 different parts of your laboratory. So the way the workflow works is we have a pre-PCR robot that can do normalization, fragmentation, cleanup, PCR prep, enrichment, all these different types of applications. And then once they're done, these specific customers prefer their own specific thermocycler. You may not have an on-deck thermocycler, so they'll take that plate manually, put it in their thermocycler, and after that PCR run, they take it to the next liquid handler, put that same plate in the next liquid handler, and it takes care of the rest. Basically, the post-PCR cleanup and the elution, and even the normalization, if you guys want to do normalization after the library prep. So this is a very common setup that we see all the time. Scaling up a little bit, last case study here is there are a lot of laboratories that have extensive liquid handling experience, a huge laboratory, and they process something like 500 samples a day, right? This specific lab is doing CRISPR gene editing for 500 samples a day. And they already have a very integrated laboratory system with rails and robotic grippers and tons of different thermocyclers on the lab. So it's almost completely automated from beginning to end. But their challenge is that the challenge is primarily space, right? They want to add additional functionality to their workflow, but they don't have a liquid handler for it. And their large liquid handlers are already doing the complicated steps of this CRISPR workflow. And they more need a smaller liquid handler to do the intermediate steps. This liquid handler needs to be integrated into a LIMS system because data integrity is very important for these customers, right? So this is kind of the setup that we usually would recommend for a customer like this, right? You have your medium or small benchtop liquid handler, and you need to make sure that, you have barcode scanning, you have LIMS integration in this liquid handler. And usually, it does the intermediate steps of the process. You have an external robotic arm that'll take it to the high throughput liquid handler. The high throughput liquid handler also has barcode scanning. LIMS integration is it's tied into the same network. And with additional robotic arms or the same robotic arm on the LIMS system, you could take it to your own PCR machine and even store it in once your experiment is done. So this is a end-to-end completely automated process that is possible. And even those smaller medium benchtop liquid handlers can easily fit into this. It's very common for a customer to have a huge liquid handler and they tell us, well, we need something to do, just the sample prep. We need something to simply take things from a 50 mil conical tubes and put it into a 96 well plate. And that's when small to medium benchtop liquid handlers shine, because a lot of people have the high throughput liquid handlers do the very complicated steps of the process, like end-to-end library prep or end-to-end um, CRISPR gene editing, right? Processes. Cool. I think that's it for content for you guys, but just moving on, I just wanted to remind you all, if you guys are going to SLAS, so SLAS is coming up in a week or so. It's going to be the biggest automation convention of the year. Very excited for it. I haven't missed an SLAS in 6 or 7 years. We will be there, and we're going to be showcasing our proteomics workstation, some NGS workstations, as well as some software solutions that that may be beneficial, right? So this is kind of what we have to offer at SLAS. We have a new product unveiling on the first day on the 27th, and we did release a teaser on social. Let me just show you guys this teaser. I'm very excited about this product and commercializing this product. See if this works. Yeah, so that was a little quick for those of you who missed it, but this is the new product right there. And we are basically launching it and announcing it on the first day of SLAS. So keep your eyes out on social. If you're at SLAS, we'll have an official unveiling. We also have a couple of very exciting talks. So this last year, OpenTrons has been working on a generative AI solution for writing protocols. So with the big boom in AI and AI being more streamlined nowadays, we actually use generative AI now to convert natural human language to full end-to-end optimized protocols. So we have two talks, one that's basically an introduction to generative AI and showcases the OpenTrons AI model on the 27th, and then a more complete talk talking about the future of automated science, generative AI, no-code software, and our modular hardware on the 28th. So be sure to stop by. We also have a poster session talking about genomics and next generation sequencing, and we have a plethora of in-booth talks. So be sure to keep your eyes out on social media and our website, and we have a ton of exciting things happening at SLAS this year. So hopefully I'll see you there. Great. I think that's it. Go ahead if you have any questions, go ahead and put them in the Q&A section. There's a Q&A box at the bottom, and you can write your questions there, and we'll use the rest of the time to answer questions. All right. So an anonymous, does OpenTrons Flex have HEPA filter / UV capability built in or as an additional module? So that's a great question. So for those of you who don't know about our concept for our liquid handlers, we're known for our industry disrupting price point, and we're known for our modular hardware and software and our open source software. The HEPA UV specifically is an easy to install module that you can purchase in addition to the base unit. And it's as easy as getting two people and installing it at the top. And it has both HEPA and UV capabilities. Next question. If you have a flexible OpenTrons in your lab, then during scientific downtime, is there a protocol for it to clean tips? So specifically for OpenTrons, we don't have a specific solution to clean tips. We don't offer metal tips. There are other providers that offer metal tips and have cleaning stations. We have had OT2 customers in the past clean their tips with an ethanol wash that they have, and it is pretty much a custom solution where they would do a bunch of aspiration and dispense steps to clean the tip with ethanol. That is an ethanol bath on the work table. So it would have to be a custom solution. Is there any conference in Europe? OpenTrons will be there. We have actually a sales team and a marketing team in Europe, and we have a lot of visibility in Europe. Europe actually is quite popular. OpenTrons is quite popular in Europe. So we do have our own sales team and our own representatives that can help you absolutely. Can you provide a rough cost of entry level to high-end unit costs? So I did cover a slide. I can go back here. Just to give you a reference of how much it would cost for an entry-level liquid handler. We offer two types of liquid handlers, one being the OT2, which you see on the left side right here. Let me actually go to this one. The OT2 being right here. And there's also, competitors like the Integra Assist Plus, which is a simpler, lower level liquid handlers. And these type of liquid handlers go from $5,000 to $20,000. Our base unit cost for the OpenTrons is, I think, $8,000 or $9,000 for this one, for the base unit price. And then high throughput large systems are definitely the more costly ones. Right here, I say between $80,000 and $600,000. But I would say if you want a fully decked out high throughput system, you're exceeding $250,000 when investing in a high throughput system. Does any liquid handler include an auto centrifuge feature? Auto centrifuge features are actually quite rare. Although I can't say that no liquid handler has it, I would say it's a very, very rare feature, and a lot of liquid handlers do not have it. But I'm not sure if there's a specific liquid handler that specializes in it. I may have seen some. A great example is something called the Kaya Cube from Kyogen. However, they're a closed system and they do very basic workflows, but I think they do have an incorporated centrifuge, but it's a very rare feature for automatic centrifugation. What is the OpenTrons? What is the options to install the modules in Europe? Is there a list price for Europe? How much can the modules of high throughput could be customized for different applications, proteomics, metabolomics, etc., with possibility of being coupled to chromatography? This is a great question. So if you're talking about specifically OpenTrons, you can access our pricing through our distribution channels. We also have our pricing available on our website. We're very transparent about our pricing, so you can go to opentrons.com right now and look at our different product catalogs, our base unit, and etc., and see all of our pricing. It is quite lower compared to our competitors, but we do have European distributors that can help you, and we have specific workstations for specific applications. Workstations meaning bundles. So if you're planning on doing proteomics, metabolomics, enzyme digestion, I would consider looking into our proteomics bundle that has, a magnet for any on-bead SPT digestions or protein purifications that you may have, as well as if you wanted to scale in high throughput, like a 96 channel head, a heater shaker for your ELISAs and stuff like that. So we are well into the proteomics space, and we do help, and we do have a provider for Europe. Are you a one-stop shop? As in you provide full IQ, OQ, PQ, and routine maintenance. So we do have a plethora of services you can purchase along with our instrument, and we are a one-stop shop. You can purchase our services, and we do preventative maintenance. We do an IQ, OQ with the plate reader on site on a yearly basis. So we do provide those services, yes. How do you ensure the validity of each module's performance, such as pipetting precision and accuracy? So we do our pipette testing and our module testing, and we have different sort of QC measurements we do before commercializing each module. So they are pre-qualified and pre-optimized. However, if you want to ensure this quality stays accurate over time, we recommend investing in our services, because we do something like an IQ, OQ for your pipettes, as well as temperature measurements year to year to see if your modules are operating at 100%. So I would say the best answer is to invest in our services, something like OpenTrons Care Plus, that that comes with preventative maintenance. Do you have ongoing roadmap for end-to-end workflows in cell culture? For now, it looks like it's more focused on proteomic and molecular and genomic assays. So for cell culture, this is a space that we have been getting into. Our product team is well aware that we need to support the cell culture space a lot more. And we do have a lot of exciting things coming in the years to come. So I guess my best answer is it is on our minds and we are considering it, so please stay tuned. Does OpenTrons have any handlers that can handle other things than tips and plates? For example, I have a circular carrier and want to pick up and place the bottom of a 24 well plate. If not, can such a system be designed? Yeah, the answer is yes. Because our software is very open source, we do deal with a lot of integrations a lot and a lot of custom solutions a lot. A great example is somebody strapped a camera to one of the gantries and they did specific colony picking applications. This is not something that we provide, but it is a custom solution that we supported. But commercially, we do not have a solution, but custom solutions can be implemented. The API of the OpenTrons machine is open, I guess. We would like to program a middleware to connect the OpenTrons robot with an external arm, for example. This is extremely possible. If you would like to learn more about our integrations and how you can integrate external arms, they do it all the time at OpenTrons, and we can connect you with our integrations expert, as I am not an expert in that, but we have that solution, or we deal with that a lot, external arms and whatnot. Which make/manufactured should we be looking at for a high throughput automated liquid handler for lots of trays and incubators? So there are the most popular high throughput liquid handlers on the market is obviously Hamilton and Tecan. They do offer tons of solutions, trays and incubators. Depending on your throughput, there are also tons of medium throughput liquid handlers like the Flex that can also accommodate your needs depending on the type of throughput. If you're talking about 500 to 1,000 samples a day or something like that, I definitely would go for those high throughput liquid handlers. And maybe something like here on the right side, right? These high throughput liquid handlers is definitely probably the best solution for you if your throughput is through the roof. We're looking for high throughput options. Does OpenTrons have it? How long do you service it for? And how is warranty wise? So it comes with a free 1-year warranty. We service it for as long as you want, depending on your service plan. We do offer annual service plans. And we do have high throughput options, obviously not through the roof, high throughput options. The OpenTrons Flex is a medium benchtop liquid handler, but we are making strides to reach high throughput. I guess the best answer I could tell you right now is stay tuned for SLAS because it will help you with your high throughput solutions, actually. So with that product unveiling. Hello, nice talk. Do you have do you recommend a way of involving stirring solution for the OT2 deck? A stirring solution, that's a very that's a great question. So because you can integrate different types of modules or different types of hardware on the OT2, if you are specifically talking about a magnetic stir, I'm sure you can incorporate a magnetic stir module or magnetic stir station into the OT2, but this would be a very custom solution. If you're trying to stir plates and stuff like that, we do have a heater shaker that can be, sorry, we're running out of time here. That could be integrated into the OT2 and the Flex. We sell it on our website. So if you're trying to stir plates or trying to stir tubes or something like that, please look into our heater shaker module. For an ELISA, you do need an absorbance reading. Does OpenTrons have a reader as adding functionality? How expensive are adding functionalities to an automated liquid handler? I would say adding functionalities to the general population of automated liquid handlers are quite expensive, but OpenTrons is known for its industry-leading price point. For example, for an absorbance reader, we actually released an absorbance reader that is an easy to use, easy to install module that you can place on the OpenTrons Flex. And I think the price is about $11,000 for that, which is quite affordable compared to other integration projects. So yes, we do have an absorbance reader. Be sure to check that out on our website. Do the units provide a detailed report of all actions completed in the workflow for traceability purposes? Absolutely. We do have log files, and it'll showcase every single action as well as any errors that occurred during the workflow. Do you have a webinar where you show OpenTron's capabilities for proteomics? Can you send a link? Yeah, I can go ahead and send that link directly to you, but we do have a series of we actually had a very recent proteomic webinar, I think showcasing protein quantification. And that if you go to our Knowledge Hub on our OpenTrons website, it's actually showcased there if you don't want to wait for the link. But we definitely have webinars that showcase proteomic applications. How does OpenTron's tip disposal unit work? So you have two options for the OpenTrons. You can either, we have a general tip waste on the top right of the instrument, and that holds a certain amount of tips. But if you want to increase that capacity, we actually have a waste chute that extends outside of the unit and slides it into an external trash can. So we have a couple of different options. Check that out on our website. Can an OpenTrons open and close Falcon tubes? Grip it to unscrew it and or seal trays and then remove the seals. So in specifically tubes, we're trying to work on a solution for opening and closing specifically tubes. Our gripper does not do that as of now, but people have integrated capper decappers into our OpenTron solutions. In terms of trays, the only option we have for sealing trays or plates is that we have our disposable PCR lids. So the gripper can seal and unseal PCR lids or PCR plates with a disposable seal with the gripper. And this is a plastic type of seal. Do you have any recommendations for periodic maintenance that should be done in order to extend the life of the liquid handler? Yes, so we do, if you look in our documentation, if you specifically are talking about OpenTrons, there is, well, every liquid handler usually has some sort of preventative maintenance form that takes you step by step on what you can do on a daily, weekly, monthly, and yearly basis to extend the life of your liquid handler. And this could be completely manual. We also specifically offer services to do this for you on a yearly basis, basically cleaning the instrument, doing the pipette verifications, and the whole suite, which is a service we definitely offer, but we do offer documentation to do some of those things yourself as well. So I would say look into preventative maintenance forms. Every company has one, and some companies even have software that will basically take you through the preventative maintenance step by step. How many systems do you have placed globally? So we have tons of systems. I would say to give you a good idea of the amount of systems we have placed globally, in this past year, we've placed close to 1,000 systems in this past year. I would say it's a little less than that. But I would say that's a ballpark value, but it's definitely in the thousands of OpenTron systems. Specifically single cell proteomics. So we do have a couple of single cell proteomic application notes that you can look at on our website in our Knowledge Hub. So we do have customers doing single cell proteomics, and we are releasing a workstation actually that highlights that in the future. Do you plan to insert any AI-powered debugger within the app before running the protocol? That would be wonderful if you can do it. We're actually currently in the process of implementing AI-powered debuggers into our protocols and run simulations. that's still a work in progress, but our AI-powered integrations are rapidly growing. I would say by next year, this time, AI will be a big part of the OpenTron software experience. How difficult is it to set up the liquid handler and software? Are there ready-made protocols or is there software programming required? Great question. So definitely, I would say that the OpenTrons is one of the easiest pieces of equipment to set up when we're talking about liquid handling. I mean, compared to other benchtop liquid handlers, it comes in a crate. And usually when I do my training for new trainees, it takes them, less than 20 minutes to set up the robot and turn it on. When we're talking about ready-made protocols, we actually have something called the protocol library on our website, and it's filled with optimized protocols for a series of different applications, proteomic, genomic, anything you can think of, that have been optimized by our applications team and our science team. So they're pre-optimized protocols that you can either send directly to your robot via Wi-Fi or download it on your USB drive and connect it to your robot. But yeah, we have a ton of different protocols and go ahead and check them out if you go to protocol library on our website. Can the OT2 be used to automate ADME testing? So I would say yes. I would say that the OT2 compared to the Flex has less functionality because of all the different modules that we have. But if you're talking about specifically the liquid handling portion of ADME tests, you can definitely do it. It may require some customization. Adding functionalities are only available for the OpenTrons Flex, not the OT2. So some of the modules are available for the OT2. The on-deck thermocycler, the heater shaker, the temperature module are all features of the OT2, which is our lower level unit for simple protocols. If you wanted any more added functionality, we also have a HEPA UV for the OT2. But if you want any added functionality like a robotic gripper or a 96-channel head or a plate absorbance reader, you would have to go with the OpenTrons Flex. I would say the price differential between the two is not that much in terms of the base unit. Great. We're 2 minutes over. I did go through all of the questions. Yeah, but that pretty much does it for today's OpenTrons webinar. Thank you all for attending. We actually have a couple of questions. Let me just answer these real quick. Will you provide a liquid handling protocol for larger tubes on the OT2 that doesn't rely on liquid sensors like the OpenTrons Flex? So in terms of OT2 support, we are currently not supporting liquid tracking. The OpenTrons Flex does support liquid tracking because it does have those type of capabilities. It does have pressure sensors and conductivity, while the OT2 does not. What is the accuracy of pipetting? Could it go into a 2mm slot? Yes, it can go into a 2mm slot, and I believe the specs on our website. Yeah, you can look up the specs on our website. I can't think of it off the top of my head, but it absolutely can. If you're talking about like digital PCR and stuff like that. Okay, I think that's all the questions I'm going to take for today. If you have any more questions, we'll answer them offline. But again, thank you all for attending. Yeah, and again, this will be recorded and sent to all of you. Yeah, and thank you for attending. Have a good day, everybody. Bye.